My name is Nicholas Blackburn. I'm at the Menzies Institute for Medical Research at the University of Tasmania in Hobart. Interesting fact about myself is that I have a cat named Zontli who was found on the side of the road in Texas and now lives here in Tasmania with me and my partner. He's named for the Aztec god of the road, Zontli, because he was found on the side of the road. And he certainly has a godlike personality in our household. So I was inspired to get involved with MS research by a colleague actually who saw that my skills in human genetics and in analysing biological data could potentially be used to make some really valuable insights and progress in the MS field. So that was my initial inspiration into MS, but certainly what keeps me inspired is the people that I work with. Not only the other laboratory researchers here at the Menzies, but also the consumers that we partner with, people with MS that tell me about their stories and their journey with MS. It keeps me fueled up to keep going with our research program here. My excitement for MS research currently goes across two areas. One is the involvement of consumers, and it's certainly not unique to MS research, but I think it's particularly powerful here in Australia, where consumers are an integral part of the research program, both as partners and researchers in their own right, joining the research programs and influencing and deciding the strategy for MS research in Australia. The other thing that excites me about MS research is our ability to draw on increasingly large international data that is being generated across from people with MS all the way down to the individual cell level biologically that we can use to integrate to find out interesting things about multiple sclerosis. So my current project is about families with multiple sclerosis. I use genome sequencing technologies to read the DNA code of people with MS in families. So I look at families with 3 or more people with MS that are closely related. So I think it could be mum and 2 of her adult children with MS, or 3 siblings affected by MS. And we read their DNA code and we look for changes to that code that are shared between every person in the family that has MS, that's not present in their unaffected family members, and that it's very rare in the general population. These DNA changes lead us to genes, and that is the biological unit that we can study that can help us understand how MS might be developing in these families. So my research is important because we do not completely understand what the cause of MS is and why it develops, nor do we understand the variability between people with MS in how the disease progresses. So my research seeks to identify the genes, the basic unit of biology, to understand how these genes influence MS risk and also MS progression at a biological level. Working in the laboratory, so for me, the laboratory is both the lab with white lab coats, but also my laboratory is this laptop in front of me as an analyst. I sit between both worlds. So I'm involved in recruiting members of families with MS, to sourcing their DNA through a blood or a saliva sample, through to extracting it in the lab, to getting that DNA sequenced and then analysing it here on a compute cluster. So some of the the most exciting bits about that for me is getting to interact across the full spectrum of that. Talking to the family member, getting their sample, and then turning it into an analysis and a result that will hopefully reveal something really insightful about that person's MS. The real challenge is just finding enough time for all the exciting aspects of this research, whether it's recruiting new people, working with my students, or analysing the latest data that's come in.